Hi guys, so today we're going to talk about the structure of DNA and talk about the different parts of DNA. Next time we'll talk about how DNA is replicated, how it leaves the nucleus, and then how it makes proteins. So what is DNA? DNA stands for deoxyribonucleic acid. It is the genetic material of life. You can also think of it as having a blueprint for life or a recipe of how we're going to make organisms. It contains all the instructions to make that organisms. And it's also found inside the nucleus. The shape is what we called a double helix. A helix is kind of a twisted staircase, and then double means there are two. And we also call it a nucleic acid polymer. So DNA is made of nucleic acids, and polymer means it's many of these molecules that are joined together. So it's a fairly large, long molecule, not just a simple one made of one or two monomers. All right, so the nucleotides are the monomers of DNA. They're going to stack up on top of each other. They're composed of three different parts. We have the deoxyribose, and remember, anything that ends in ose is sugar. We have the phosphate, and these again are going to have a slight attraction. There's the phosphate. And then we have one of four different nitrogenous bases. So we're going to talk about the different nitrogenous bases. So the nitrogenous bases are the molecule that contains nitrogen and have the chemical properties of a base, meaning a base is the opposite of an acid. There are four different ones we're going to use, only four. We have adenine, which we use A, guanine, so we denote that as G, cytosine, that's C, and thymine, which is T. So these are the four bases we're going to use to build our ladder of DNA. So here we have our sugar attached to our phosphate, and then here we can have our A attached to the ribose, or we can have our T, or we can have our G, or we can have our C. Now these are going to attach in very regular ways. So I want to talk briefly about purines versus pyrimidines. And you're not going to be responsible for these on the test, but I do just want you to kind of think about the difference between the two. So we have the purines and the pyrimidines. The purines are the A and the G. They're larger, okay? And they contain two rings. So remember, we talked about how a lot of these ring structures form in organic molecules. So if you look, you have two rings. Adenine has two rings and guanine has two rings. These are purines. Then we have the pyrimidines. These are smaller and they only have one ring. Our pyrimidines are the C and the C and the T. Okay, so you can see there they are simpler and they are smaller. So how do we put our DNA together? Well, we have to make our twisted ladder. And on the side, we have our ribose sugar, that's the pentagon, with the little circle on the side, that's our phosphate. And then we can attach any nitrogenous bases to one side of the ladder. Now, the great thing is once we know one side of the ladder, we also know the other side of the ladder. All right, so let's load up our picture. So you can see that once we know that C is on one side, we know that G is on the other. T pairs with A, G pairs with C, A pairs with T, over and over again. So these are always going to be in anti-parallel, meaning they run in opposite directions. And I'm not going to do too much with the priming ends. All right. Just know that they are anti-parallel. So the rungs of the ladders have our nitrogenous bases. The rung is like where you would put your hands and feet if you were climbing a ladder. And we always are going to use our base pairing rules. So A always pairs with T, and G always pairs with C. And the way that I remember it is at. If I can spell the word at, then I know where my DNA is at. Okay? So A pairs with T and C pairs with G. All right, so adenine and cytosine would not go together, right? That does not spell at. Guanine and thymine would not go together. Nope. All right, these are held together by relatively weak hydrogen bonds. So remember when we talked about water, we talked about properties of water and how a water skater can skate across the water due to the hydrogen bonding, breaking and reforming. Well, DNA, the nitrogenous bases, are also held together by this relatively weak hydrogen bonding. And what this allows the DNA to do is be able to split open and reform relatively fairly easily. If you notice here, if you look between the A and the T, we actually have 2 hydrogen bonds. And C and G, we actually have 3. So if you know one side of the letter, you know the other. So let's do some base pairing practice. T pairs with A, C pairs with G, C pairs with G, G pairs with C, A pairs with T, G pairs with C, and T pairs with A. So again, once we know one side, we can fill out the other. And you guys are going to be practicing this in class. All right, that's all I have for you today, and we'll see you in class tomorrow.